Headlines are calling this a new class of anti-aging drug, but here's the reality. A lab study in yeast used a next-gen mTOR inhibitor called Rapalink-1 and uncovered a clever link between a gut-related molecule and the cell's aging switch. It's a neat mechanism, not a human anti-aging pill, at least not yet. mTOR is the cell's growth and nutrient-sensing hub. When it's cranked up, cells favor growth. When it's dialed down, cells tend to prioritize maintenance and repair. Across many lab organisms, gently lowering mTOR activity is associated with longer, healthier cell life. Rapamycin made this idea famous. Rapalink-1 is a more engineered cousin in the same family that lets researchers probe the pathway with extra precision. Here's what the study found in simple terms. A molecule called agmatine, something that can come from diet and gut microbes, feeds into an enzyme called agmatinase. That chemistry appears to influence TOR complex 1, the mTOR complex tied most closely to nutrient signaling. In the yeast model, that agmatine to agmatinase route helped shift TOR complex 1 toward a lower tone, which lined up with better cellular housekeeping and longer lifespan in that system. What this does not mean, we did not just discover a proven anti-aging drug for people. Yeast are powerful models for mechanisms, but they are not humans. Also, Rapalink-1 isn't brand new to medicine. It's part of the broader mTOR inhibitor landscape that's mostly being explored in areas like cancer and transplant medicine, not as a longevity supplement. And no, this is not a green light to rush out and buy agmatine. The cautious takeaway is mechanism first, human evidence later. Why it matters is the bigger story. This adds weight to a growing theme. The microbiome and metabolism can tune mTOR, which shapes how cells balance growth versus repair. If this agmatinase signal shows up in mammalian cells or animal studies, we'll have a new handle to test, and maybe one day to target in a clinical context. So what can you actually do today? Don't chase shortcuts off a yeast paper. Stick to habits that are already shown to nudge cellular maintenance in the right direction. Consistent exercise, adequate sleep, smart nutrition, and periods of caloric balance aligned with your goals. Then, watch for the next step in the evidence chain. Replication in mammalian systems, biomarkers that confirm TOR1 is moving the way we think it is, and safety data that shows we're not trading immune function or resilience for theoretical gains. New anti-aging drug makes a great headline. The real story is smarter, a microbiome to mTOR lever that helps explain why certain diets, microbes, or metabolites might influence aging, at least in yeast for now. If you want me to follow this into mammal data and break it down when it lands, hit subscribe and drop your microbiome longevity questions in the comments.